pleasure to introduce Dr. Preeti Dalawari, who will formally introduce our speaker today. She earned her Bachelor of Science from Penn State and MD from Jefferson Medical College through a combined program, then completed her residency in emergency medicine and a Master's of Science in Public Health and Epidemiology at St. Louis University. She spent 15 years at SLU as an associate professor in emergency medicine, where she served as division research director, taught countless students and residents, published over 25 research articles, and edited a text on the intersection of social determinants of health in the emergency department. Since 2020, she has served as the vice president and medical director of U.S. Individual Life at Reinsurance Group of America, and as managing director of the Longer Life Foundation, a collaboration with WashU that supports research on health, wellness, and longevity. Please join me in welcoming Dr. Dalawari.
Good morning, everybody. Thanks for being here for Grand Rounds. I know this is something you guys do weekly, but from the Longer Life Foundation aspect, it's always a pleasure to be here to be able to look and hear about research from our former grantees. So the Longer Life Foundation is, as Dr. Danier mentioned, a collaboration between WashU and RGA, RGA being one of the larger life reinsurers in the country. And our sole purpose is funding research for morbidity, mortality, longevity. We just completed our cycle for the 2025-2026 year, and I am very happy to congratulate our newest grantees, which are Dr. Monica Bambuskova, Dr. Rita Brookhart, Dr. Ali Reza Ganbarpur, Dr. Eva Klinman, Dr. Laura McPherson, Dr. Joshua Oltmans, Dr. Max Peterson, Dr. Priyanka Verma, and Dr. Lindsay Underhill. We go through the cycle every year. It's going to start again in January, so if anyone's interested, please reach out to me or reach out to Dr. Reeds. And I will hand it off to Dr. Reeds, who is the director of the Longer Life Center, which is basically our support on this side of the I-64. Thank you.
Good morning. I promise I'm going to be the last person introducing someone this morning. So we are lucky enough to have Dr. Nate Stitziel with us today. He's a professor of medicine and genetics and associate director of WashU's MSTP program. He's taken somewhat of a circuitous path here, which I always think is quite good fun. He started off as a math and physics major at WashU, and then discovered that he could actually do medicine and see patients some of the time and do some research the rest of the time. So with that in hand, he received his MD and PhD degrees from the University of Illinois and completed residency at University of Chicago's PSTP, followed by fellowship in cardiovascular medicine at Brigham and Women's and a postdoc research fellowship at the Broad Institute and Harvard Medical School. So as you can see, pretty poorly trained before being recruited back to WashU in 2012. Since then, he's directed a multidisciplinary basic and translational research group focused on understanding the inherited basis of cardiovascular disease to identify novel targets. In recognition of his work, he's now been elected to the American Society for Clinical Investigation, the Association of University Cardiologists, and was recently named a 2025 Harrington Discovery Institute Scholar Innovator. So kudos to you. So without further ado, Dr. Stitziel.
Okay, thanks everybody. Thank you for that very kind introduction, and thank you to the Longer Life Foundation. The foundation's been helpful for my research and others, and it's foundation funding really that gives us support to do work that is not traditionally fundable through traditional grant mechanisms. These are the disclosures that I've been asked to show. I'm going to start with, I think, what the most important slide is, and so these are the people that I get the fortunate day-to-day chance to work with, both present and past. These are the incredibly creative and passionate group of scientists that I feel really lucky to work with. And so, I like to make the joke, like when I say that I came up with something, it's really like we came up with something. And then when I'm up here and I say like we did something, it's like they did something. And so these are the people that do all the work. I get, to stand up here and talk, but again, I'm really lucky to work with these people. So I think that we can all agree that there's a problem, which is that there's a persistent need for novel therapies targeting cardiovascular disease. We haven't solved this, right? We've been working on it for a long time, but there remain a need. And it's a nested problem. So the problem why we have a persistent need is that there's a really high failure rate of cardiovascular drugs. And I'll tell two quick stories to sort of highlight this. Before I do that, I will highlight the numbers. So this is a review from a little over 10 years ago, looking at the percent success of agents that are either entering a phase 2 or phase 3 trial. And in the cardiovascular space, you can see here, of agents entering a phase 3 trial, think about all of the time, money, effort it takes to get to a phase 3 trial, half of them end up succeeding, getting approval, which sounds good, but that means that half don't succeed. And if you look at the likelihood of approval from phase 1, there's 7% of agents entering a phase 1 first-in-human trial that end up succeeding. 93% of agents are failing. And I don't know, there are probably lots of reasons. I can think of at least 2 reasons. Maybe you can think of others. Maybe it's really difficult to target those targets, right? Maybe you've got the right target and you just can't drug it. Or alternatively, maybe you can do a really good job at targeting what you're targeting, but you're just hitting the wrong target. The first vignette I'll tell you is anemia and cardiovascular disease in kidney disease. And there's been this long-standing observation that individuals with kidney disease that had anemia had higher proportion of cardiovascular disease. And so there was lots of effort to try and raise blood counts in these individuals. And Mark Pfeffer, when I was a fellow, led this trial in which they answered the question to whether or not raising hemoglobin in these individuals would result in benefit. It turns out there was no benefit to raising hemoglobin in these individuals, and in fact, there was harm. And so these individuals had significantly higher rates of fatal or non-fatal stroke. So the drug worked. It did what it was supposed to do. It was just the wrong target. And then there's this story, of course, of HDL and cardiovascular disease. So there's a long-standing observation that there's higher rates of cardiovascular disease in individuals that have very low HDL cholesterol. I asked ChatGPT how much money has been spent on this, and it said $5 billion on the HDL hypothesis. It works, right? We can do this. We can raise people's HDL. This is one agent of, 10 agents or more that raise HDL. It just doesn't work, right? It just doesn't work, no matter what you do. So again, the chemistry works. The chemists are really good at making drugs that they're supposed to do. It's just the wrong target. And this is actually borne out in this root cause analysis from these authors, right? Where they look at why were these drugs suspended. So in the blue bar, of drugs entering phase 3 trials, half are suspended because they just don't work for the disease that they're being developed for. They're not effective. It's not a safety concern. It's not a chemistry concern. They just don't work. And so I'll argue that there's probably lots of reasons why this happens, but at least one reason is that we've been historically working with insufficient models of human disease, in which we rely on epidemiologic observations or animal studies to identify therapeutic targets and pathways. We spend a lot of time and effort to get to the point where then we can actually finally do the trial in humans to see whether or not it works. And it turns out that there's lots of issues with determining causality from epidemiologic studies, and things that cause disease in animals, of course, don't always cause disease in humans. So how can we improve? As you might imagine, as a human geneticist, us and others have made the argument that we can use human genetics to try and start with human genetic loci to understand disease patterns and processes, because if we start with humans, more than likely, it's going to end up being beneficial. And so the model is that we try and understand the loci that underlie disease. Of course, there's always room and need for mechanistic studies in animals. You can do mechanistic studies in humans as well, as I think we'll argue. And then there's ways to use human genetics to then try and validate the therapeutic targets that end up coming out of those studies. And of course, like in academic medicine, there's no new idea, right? Every idea is like an old idea. So this is Paul W. White, who's considered the grandfather of cardiology, one of the first cardiologists. And he recognized this from a long time ago, that the individual and the background of heredity are just as more important as the disease itself for diagnosis, prognosis, and treatment. And again, it doesn't take Paul W. White or some master clinician to understand that there must be genetic loci that underlie disease. You guys know Bob Odenkirk from Better Call Saul. For the aficionados in the audience, any Mr. Show fans out there? Mr. Show, Mark likes Mr. Show. You may remember that Bob Odenkirk had a heart attack, like on the set of Better Call Saul as they're wrapping up. And so, relatively young guy, seems healthy, has a heart attack. And then personally or anecdotally, I think we all know people like this, right, who just can't develop disease no matter what they do. And so, the question that our lab and of course many other labs are focused on is like, can we leverage the misfortune of Bob or the fortune of Aunt Betty to try and understand what's driving disease, and can we use that to identify therapeutic targets? And so the generic sort of like 30,000-foot view of a genome-wide association study, right, is that you take a bunch of people like Bob and a bunch of people like Betty, and then you try and figure out where do they differ in the genome. And this has been done by us and others, and this is a review from, 10 years ago now, showing a Manhattan plot. If you've been paying attention over the last, 15 years, you've probably seen one of these. And so the point is that there's lots of genetic loci that are robustly associated with disease. These are genomic spots, right? These are like signposts for things in the genome that are causing disease. And for coronary disease, there's actually now over 250 loci that have been identified statistically very robustly. But we actually don't know the causal gene at any, we know a handful maybe, but we don't know the causal gene at many of these. And so the question is, how can we move from locus to gene? And so I'll tell you the results, sort of top-line results of one of the very first things that I did when I first came here, which was to lead a very large genome-wide screen for protein-altering variation associated with coronary disease, with the idea that if a genetic variant is altering a specific protein or a gene product, then maybe that's going to be a hook into trying to identify things that are associated with disease. And it worked in the sense that the top results were LPA, which encodes lipoprotein A. Certainly looks like a causal trial or a causal factor. We'll find out with phase 3 trials that are ongoing. And PCSK9, which is, of course, as you know, approved for lowering LDL cholesterol and treating coronary heart disease. The first novel result that I'll highlight is ANGPTL4, which seemed to work through triglycerides. Even though this is 2016, you may or may not believe it, triglycerides were sort of still in the debate, HDL and triglycerides, which one is it? And there was some uncertainty as to whether or not triglycerides were in the causal pathway. But this was associated with a protein-altering variant in ANGPTL4 was associated with coronary disease at a genome-wide level of significance. It was known to be a partial loss of function, but there was some debate in the literature as to whether or not this was certainly associated. And ours was the first definitive association with coronary artery disease. And interestingly for us, it significantly altered triglycerides, but it had no impact on LDL cholesterol, sort of giving a hint that triglycerides were probably going to be in the causal pathway. ANGPTL4, if you don't know, is an endogenous inhibitor of the enzyme lipoprotein lipase, which is the main way by which triglyceride-rich lipoproteins are hydrolyzed in the circulation. And we were able to take protein-altering variants across a range of LPL activators or inactivators, like ApoC3, ANGPTL4, ApoA5, as well as LPL itself. And we were able to demonstrate that genetic variation that served to reduce LPL activity had an effect of increasing triglycerides and raising risk of coronary disease, and the converse. So genetic variation across these agents, or across these molecules that increased LPL activity, all served to lower triglycerides and reduce the risk of coronary artery disease. And so we made the argument that this suggests there's an allelic series that modulates enzyme activity, triglyceride-rich lipoproteins, and coronary disease. It suggests that LPL and TRLs are in the causal pathway for CAD, and because there's multiple things that modulate LPL activity, it increases therapeutic opportunities across the entire pathway. Luca Latta, in separate work, then validated this LPL activation and risk for coronary disease in a genetic study in which Luca took individuals, a large number of individuals, and looked at genetically lower predicted triglycerides specifically via the LPL pathway, and was able to demonstrate that individuals who were predicted to have lower triglycerides specifically via the LPL pathway have reduced risk of coronary disease. And then Luca went on to subset those individuals into people who were predicted to have genetically higher LDL or genetically lower LDL, and you can see, I think, that it's additive. So LPL pathway and LDL metabolisms appear to be sort of additive pathways for protection against coronary disease. So an ideal agent may be one that actually targets both of these. And it turns out that there is such a molecule, and it's ANGPTL3, which you may have heard about. So ANGPTL3 is a circulating molecule that acts as a lipase inhibitor, including LPL. There is a monoclonal antibody that's approved for homozygous FH called evinacumab. There are other agents in development. It lowers triglycerides via LPL activation, and it lowers LDL cholesterol via a, I think fair to say, yet to be ascertained mechanism. There's lots of other examples, however, where genetic loss of LDL or genetically lower LDL leads to increased hepatic fat, and there was some concern, I think, that this may be similar for ANGPTL3. And this was highlighted by this agent, vupanorsen. This is a clinical trial looking at vupanorsen, which is a hepatically targeted antisense oligonucleotide against ANGPTL3, in which the individual or the investigators identified a dose-dependent increase in hepatic fat. And so then there was a question, is this an on-target effect of inhibiting ANGPTL3? Should we abandon this pathway? Or is this an off-target effect of the drug? And so we decided maybe we could make a comment on this using genetics. And so in collaboration with Tyler Fromm, who's at the Mallinckrodt, and Tom Neufeld, who's currently a pediatric cardiologist but was a medical student at the time, we studied a group of individuals in St. Louis who are ANGPTL3 knockouts. They have biallelic loss of function. And then we collaborated with my friend Marcello Arca in Rome and his student Laura D'Erasmo, where there's a village called Campo de Mele, south of Rome, in which there's a large proportion of individuals that have loss of function for ANGPTL3. We stuck all these individuals in the MRI scanner, and it turns out that there really is no increase in hepatic fat over their lifetime. We made, of course, there is a caveat of, there's not that many people that have two loss of function alleles, so these are either homozygous loss of function or compound heterozygous loss of function. And so, there may be some still remaining questions as to whether or not this has something to do with hepatic fat. And so we used a technique called Mendelian randomization that I'll review a little bit later in terms of the framework, but I'll just point out that we were able to basically predict individuals' ANGPTL3 levels and look at their hepatic fat outcome in the UK Biobank. As positive controls, we looked at genetically predicted ANGPTL3 on the x-axis and its effect on plasma triglycerides. I think you can see there's a very strong effect, which you would expect biologically. It also associates with LDL cholesterol. Again, that fits with the biology, but there was really no association with hepatic fat in large numbers of individuals. I think this is 30,000 individuals from the UK Biobank, suggesting that this is probably an off-target effect of vupanorsen and not an on-target effect of targeting ANGPTL3. And in fact, there are now new agents. So this is an arrowhead agent, which is a silencing RNA. And they specifically did pre and post hepatic MRI because of the vupanorsen signal, and they did not find any increase in hepatic steatosis. So I think it's fair to say that this is not going to end up increasing risk for hepatic steatosis. And amazingly, ANGPTL3, and you can correct me if I'm wrong, I think this is the only molecule that lowers LDL even in the absence of the LDL receptor. That was first observed in mice and then followed up in humans. And so this is a small trial of homozygous FH, individuals that are given either placebo or evinacumab. You can see that ANGPTL3 inhibition substantially lowers LDL cholesterol durably over a 2-year period of time. And then they looked at the genetic reason for why they're homozygous FH. And so you can have what's called a non-null mutation in the LDL receptor. This is like a partial loss of function, or there's some residual function. The receptor's still made, it just doesn't work like it's supposed to. Or you can have what's called a null-null mutation. And so these are individuals that have 2 nonsense alleles, for example. They literally do not make the LDL receptor at all. And evinacumab durably lowers LDL cholesterol regardless of whether or not you have the LDL receptor. This is consistent with mouse data in which these knockout mice literally don't make the receptor. And I think, again, this is the only molecule that participates in this pathway, which is really kind of fascinating. So of course, we asked, well, how does this occur? What's the role of ANGPTL3 in hepatic metabolism? And I'll give you top-line results of sort of 2 studies. One was a collaboration with Bettina Mittendorfer, who was here and is now some very high-level position at the University of Missouri, along with Bruce Patterson, who's one of these mathematical gurus. for label or for doing kinetic studies. And we took that St. Louis family that I talked about, where there's individuals that are complete loss of function for ANGPTL3. We took some wild-type controls, and we looked at the appearance of labeled lipids over time, if they were either oral or IV. And then Kendall Burks, who was an MSDP student in the lab, we did a collaboration with Nick Davidson, where we took immortalized hepatocyte cell lines, and we used CRISPR to knock out ANGPTL3. And again, I could sort of give you the high-level results that plasma triglycerides and TRLs are dramatically reduced in vivo, and that there's a markedly reduced liver-derived secretion of triglyceride-rich lipoproteins. So this happens both in people and in our cell model. I told you that there's no increase in hepatic fat in the people. That actually also is consistent with the cell model. And we think that this is actually due to increased fatty acid oxidation and PPAR alpha activation. We're still working on sort of how that metabolism shifts, and if the cells are secreting less lipids, how do they not actually accumulate lipids? So that's an ongoing work in the lab. And again, I've shown you the human data in vivo that there's normal LDL uptake even in the absence of the LDL receptor, and we've now created a cell model of that to try and study how this happens. And so we took these cells that I mentioned. We have wild-type immortalized cell lines, or cell lines in which we've used CRISPR to knock out ANGPTL3, and we took the wild type and we knocked out LDL receptor, and we took the ANGPTL3 knockout and we knocked out the LDL receptor. That's shown in this protein blot here, and so now we have 4 different genotypes, one of which we call double knockout cells, DKO, where there is no ANGPTL3 and there is no LDL receptor. And we studied LDL uptake, so this is fluorescently labeled LDL in the dish, and we quantify how much uptake of LDL these cells can participate in. Compared to wild type, these are all normalized to wild type uptake. ANGPTL3 knockout had no difference. As you might expect, if you knock out the LDL receptor, you get a dramatic reduction in LDL uptake. And remarkably, in double knockout cells, you basically normalize uptake. And so this is the non-LDL dependent, or non-LDL receptor dependent uptake that we're studying. I hope you invite me back. We have a scientist in the lab, Etienne, who's made a lot of progress in the last 2 years, and I think we're sort of getting on our way to trying to figure out what is going on and how do these cells actually clear LDL in the absence of the LDL receptor. So we've had lots of agents to study lipids. It'd be great if we could identify something sort of non-lipids. And so from the same study, in addition, we, for better or for worse, had two novel results. One was this ANGPTL4, and then the other was this protein that nobody has ever heard of and you've never heard of called SVEP1, except the guys in the lab have heard of this. SVEP, we call it in my lab. No idea what the pathway was, but robust association with a protein-altering variant in coronary disease. So again, like a genomic signpost that is saying, whatever's happening here, this is important for disease. It turns out that this protein-altering variant was an aspartic acid at 2702, replaced by glycine. We still don't fully understand the molecular impact of that specific amino acid change, but ours was the first association with any human disease. There's now actually been a catalog of human diseases that are associated with this protein called SVEP1. And exciting for me as a junior guy at the time, I guess maybe I'm still junior, exciting for me, there was no impact on lipids, right? So this is a non-lipid, potentially a non-lipid pathway for coronary disease, which is really, I think, as a field where we need to start moving to treat atherosclerosis. We can get people's LDL down to 30. There's still events. We need to start targeting non-LDL pathways. Of course, I spent the first 3 minutes harping about how correlation is not causation. And so we tried to set out, is SVEP the causal gene in the locus? So how do you prove causality in humans? I think most of us can agree that the gold standard is the randomized clinical control trial. You give treatment, importantly, that's randomly allocated at entry before the cataloging of any events, right? You give drug and placebo. And so in individuals that get the placebo, whatever biomarker you're looking at, I'll use the example of LDL cholesterol. The biomarker is unchanged. It is what it is in those individuals. And then you give a drug, for example, to lower LDL cholesterol. The biomarker levels are reduced. Importantly, the confounders are equally distributed between the groups. That's why this works. So there's smokers in both groups, for example. There's whatever else in both groups. And then you figure out, is there a difference in the outcome of coronary heart disease? And so I think short of randomized clinical control trials, the only other way that I know of that we can sort of suggest causality in humans is Mendelian randomization. And the reason it works is that it's the same framework, right? So alleles are randomly allocated at birth. There is no reverse causation that we know of, at least. Like, disease is not causing germline DNA changes. So those alleles are allocated at birth before any events. And you can look at a series of alleles that either don't change the biomarker, for example, LDL cholesterol, or you can look at alleles that lower LDL cholesterol. So this is genetically lower LDL cholesterol over your entire life. And then again, you can ask the same question. Is coronary heart disease unchanged? Is it reduced? Is it increased? And so this is a review from 2020 from Steve Burgess, who was one of the OGs of Mendelian randomization. And they make the point that if you look at LDL cholesterol, like, this works, right? These are LDL cholesterol increasing alleles, coronary heart disease increases. If you look at C-reactive protein, which is a marker of inflammation, it's not driving disease, it's a reflection of inflammation. There is no association between genetically altered CRP levels and risk of coronary disease. So this is the framework of using this sort of Mendelian randomization. So we set out to ask, what about SVEP and its levels and its association with disease? And we sort of got into this because of this paper from my friend Adam Butterworth, in which they took individuals from the interval study, about 3,000 people. This is a healthy cohort from the UK. And they measured about 3,600 plasma proteins. Turns out that SVEP was one of the plasma proteins that they measured. And they looked at genome-wide DNA genotypes. And I'm literally looking at this paper, and I'm looking through the supplemental table 37. And it turns out that this protein-altering variant that we found associated with coronary disease is a substantial, it's associated with substantially increased levels of this protein in the plasma at a very strong level of statistical significance. So this got us thinking, wow, well, maybe we can actually use this approach to try and look at alleles that alter the protein levels. And so we obtained the data from Adam, and it turns out that if you do we didn't do this, but we just sort of plot the data. If you look at this genome-wide association study for SVEP levels, the strongest locus, as you might imagine, which I think is a good positive control, is the locus that encodes the protein, SVEP. And so Erica and CJ, the group, did a Mendelian randomization where they took independent genetic variants that all associated with these protein levels at a genome-wide level of significance in the interval study, and then importantly in a completely separate study, so this is like a large GWAS for coronary disease, asked if those genetic variants associated with coronary disease. Again, this is called two-sample Mendelian randomization to try and avoid any confounding between the groups. And I think you can appreciate that genetic variation that served to increase this level of the protein was associated with increased risk of coronary disease and vice versa, suggesting that this is indeed causally related to human disease. So I think I'll wrap it up. We're done. It's the causal gene, right? As a great American poet once said, haters are going to hate, and somebody's going to say, we're in Missouri, and this is the show me state. And so leave your statistical voodoo and mathematical dark arts at the door. And also, you wrote an editorial talking about the misuse with Paul in the audience about the misuse of Mendelian randomization. So I'll show you the data. So we have a suggestion that it's in humans, and then of course, we need to really sort of prove causality in mice. And so In-hyuk, who's a faculty member in the lab, led a study in which he took animals that were either heterozygous, had heterozygous loss of function for this protein, and atherosclerotic burden was reduced, or took animals in which we depleted all of SVEP from only smooth muscle cells. So it turns out that this protein's made by smooth muscle cells within the developing plaque, and those animals were homozygous depletion for smooth muscle cell specific knockout, and those animals also had a reduction in atherosclerosis. I think confirming that this is a causal factor for the promotion of coronary disease. Of course, that doesn't tell us how this is actually doing anything and how it's causing disease. At the time when we mapped this, there was an integrin alpha-9 beta-1 that was the only known receptor to interact with SVEP1. And so I'm a simple guy, and I say, hey, In-hyuk, let's talk about this. And we both decide together, well, we should knock that out. It turns out that knocking that out in mice has no impact on atherosclerosis. And so it looks like that's probably not how SVEP is interacting or influencing disease. And so we said, okay, well, there may be other cell surface receptors that this ligand is interacting with. And so Jaron Ellenbos, who was a MSDP student in the lab, so we were looking at this Manhattan plot one day, and we said, this is really weird. So this is SVAP, that makes a lot of sense. But then there's this what's called a trans PQTL, so protein quantitative trait locus, on chromosome 1. Like, that's kind of weird. Like, how is a genetic variant on chromosome 1 is influencing the protein levels of this protein that we're studying? And so we sort of started to dig into this a little bit more. And it turns out that this locus encodes a protein called pair 1. And so I'll tell you a little bit about pair 1. So pair 1 is named platelet and endothelial aggregation receptor 1. It's not a great name because it's expressed by more cells than just platelets and endothelial cells. There's also not pair 2, but these people name these proteins, I think, thinking that they're going to find additional ones. It's the strongest platelet reactivity locus in humans. There is a non-genome-wide significant, but a strong genetic association with coronary disease. It's a receptor tyrosine kinase-like protein, and amazingly, it was an orphan receptor. So it had been identified in a platelet screen in 2005, and no one knew what the endogenous ligand was for this receptor. And interestingly, the entire signal on chromosome 1 for association with levels of SVAP, which is the protein that we study, was due to a protein-altering variant in the extracellular domain of the receptor, so D343N. When we condition on this variant, the association in the locus goes away with the protein that we're studying. So that suggests that this is a really important genetic variant for the protein that we're studying. This is sort of the box plot here of individuals that carry these, the N at 343, and as you can see, it relates to higher proteins. It does not relate to levels of the protein pair, which is the receptor, so it only relates to the ligand. And sometimes we have these things with proteins where there's like proteins that can interact with all sorts of proteins, right? And so sometimes you have these, there can be a chromosome, there can be a genetic variant on chromosome 1 that might associate with 500 proteins. And so we looked, and it turns out that there is only association with that genetic variant on chromosome 1 across 5,000 proteins with SVAP, which is the protein that we study. I've harped on how correlation is not causation, so we set out to find out if these proteins actually do physically interact. And so I'll describe to you the results of an experiment which is using a technique called biolayer interferometry, in which we are able to load pair on the tip of an optical sensor. We can then expose that sensor to SVAP, which is the ligand, and we can watch a wavelength change up the optical sensor as protein associates. And so if that surface of the sensor becomes thicker, it reflects in this shift that you can see here. So this shift is an optical signal that's indicating that the surface of the sensor is getting thicker, and then we can move that sensor into a buffer that does not contain any ligand, and we can watch as it comes off. And so this gives you, a kinetics of an on rate and kinetics of an off rate, and then you can use those to calculate what's called the affinity constant, KD. I'm not a protein biochemist, but the guys in the group tell me that this is quite avid. So 9 nanomolar is on the range of, getting close to antibody avidity. And then interestingly, at least directionally concordant, the N allele at 343 led to a 2-fold lower affinity. And if you recall, we saw higher levels in the plasma. And so this really suggests that these two proteins do, in fact, physically interact. And that may be the reason why we're seeing higher levels in the plasma of the ligand. And then we went on to try and show what the impact of this association might be. Jared was able to show that these cells, when cultured on top of SVAP, were able to activate phospho-AKT signaling and downstream mTOR signaling. I won't show you the mTOR blot. But you can see, I think, that as compared to a neutral control BSA, when cells are cultured on top of this ECM protein SVAP, there's phospho-AKT signaling. This pair polyclonal antibody dimerizes the receptor, so it acts as a positive control. And then, of course, serum acts as a positive control for these growth pathways. And AKT and mTOR signaling, I think, is consistent with all of the disease processes that this ligand is now associated with. So chronic AKT and mTOR, probably not a good thing. In collaboration with Jorge de Paula, who's the chief of pediatric hematology and studies platelets, Katrina in his lab was able to do a platelet aggregation assay. And I think you can appreciate that when platelets are not exposed to SVAP, they don't aggregate, and then they nearly fully aggregate in the presence of this ligand. PAIR is, again, a very important platelet receptor for platelet aggregation, suggesting that this is actually how this works. And then we're able to demonstrate that those same signaling pathways, AKT, mTOR, were active in platelets as well when they were exposed to this ligand SVAP. And then CJ in the group was able to do Mendelian randomization for cardiovascular disease, suggesting a hypothesis. Again, this is not formally proving, but suggesting that pair levels in addition to SVEP levels are concordantly and positively associated with cardiovascular disease. So SVEP is, it is required for development. So this is not our work, this is work of others that is looking at homozygous null animals. And these animals die of embryonic lymphedema. So lymph vessels and lymph valves don't form normally in animals that lack this protein in development. But we've taken animals that have what's called a floxed allele, and we've crossed these animals with a Cree expressing allele in all cells that's inducible. And so we can induce loss of function at 8 weeks of age, and we've aged these animals out now to about 2 years, and there does not really appear to be any adverse effects. So we think this is potentially targetable. There's lots of examples of proteins that are required for development, and then due to the shadow of selection, quote unquote, become less important later in life, and in fact, when turned on, are deleterious. And I'll highlight work that Arturo has led for a couple of years now, where him and Ryan, who is a postbac and now a medical student at the time, have been really leading incredible efforts to try and drug this interaction. And so the cartoon here that I'm showing is that SVEP is the ligand that we think is important for dimerizing pair. When pair gets dimerized, it becomes phosphorylated. Then it triggers this signaling cascade of AKT-mTOR that we think leads to cell behavior and disease. And we've taken two approaches. One is to develop a therapeutic blocking peptide, and then the other is to develop a small molecule screen. So the small molecule screen worked. We did it in about 40,000 compounds, and we have a series of compounds that actually look like they actually quite robustly inhibit this interaction. We have more functional data for the therapeutic blocking peptide, and so I'll show you just one slide on the data we're really excited about, in which I think this is the blot I showed you before, in which SVEP activates this signaling pathway, phospho-AKT. When we culture those same cells, again, phospho-AKT is activating when we culture them on SVEP. When we add this blocking peptide, we can abrogate that signaling pathway, which we're really excited about. And the signaling pathway appears to be somewhat specific because when we culture cells on top of serum, there's no effect of the blocking peptide, which you would expect if this is really a specific peptide that's that's blocking that interaction. And then from a cell behavior perspective, again, we collaborated with Jorge and Katrina, and we gave them our blocking peptide. This is the same thing that I showed you before. When you expose platelets to SVEP, you get aggregation of platelets, and we're able to reduce this in a dose-dependent manner, and in fact, effectively block all aggregation that's due to SVEP at least, not due to other pathways, but certainly due to SVEP. Okay, so that's like one locus down. Of course, there's like hundreds to go, and so how are we going to like make progress on the rest of the work? So one effort that we are leading with others is discovering new alleles. And so this is a program that CJ from the group is participating in, where we're analyzing whole genome sequencing data that has been generated in a very large number of diverse individuals. So as you may know, many of the genetic association studies have been done in individuals of European ancestry, and there's a limited array of genetic variants in individuals of European ancestry. And so I think there's lots of reasons why we would want to study diverse individuals. And so we, with Paul, Tim, and Jihong, shown here, have pulled together over 250,000 cases and controls to study new alleles from diverse individuals that have been discovered through whole genome sequencing. And this was done, again, many of the samples were sequenced here from a program called the Center for Common Disease Genomics. And then you may have heard of this program called NHLBI TopMed, and then, of course, the UK Biobank, with plans to replicate what we find in the program from the NIH called All of Us and the VA Million Veterans Program. So that's one effort that we're doing to try and find, how are we going to understand, new alleles and find new ones. And then, of course, there's efforts to map genes in existing loci, right? I mentioned that there's 250 loci for coronary disease, and we know the causal gene at maybe like a handful. And why do we not know the causal gene anymore? Well, it turns out that the vast majority of these loci are located in non-coding regions of the genome. If these were all in coding regions, like I showed you the very first study, It may be easier, but that's just not how the genome biology works. But these are enriched in areas of open chromatin. And so you may know that the way that the genome is organized is that there's lots of areas of compact chromatin, and then there's open chromatin sites, which they're not bound by nucleosomes. These are called DHS sensitivity sites, DNase hypersensitivity sites, DHS. And I show that because the figure that I'll show you next uses the term DHS. And so that's what it means. When it says DHS, it means that there's an area of open chromatin. And these areas of open chromatin are, of course, important for recruiting transcription factors, which then, of course, drive gene expression, right? That's why these are important to look at, because these are regulatory regions that are active. And so Matt Morano, this is not my work, this is Matt Morano's work from over 10 years ago, mapped DHS sites across the entire genome agnostically, and then asked the question, if you look at all DHS sites across the genome, and then you look at what's called the GWAS catalog, so like all of the alleles that have been associated with any complex trait in a genome-wide association study, I think you can appreciate in the green part of the pie graph there that there's an enrichment of these GWAS SNPs in these sites that are active regulatory sites, which makes sense because if they're in non-coding portions of the genome, but they are robustly associated with disease, they're probably affecting disease through gene regulation. But we still don't know the genes. Like, even if we know the DHS sites, like, we still don't know the genes, right? Because tissues are made up of lots of different cell types, and so there's cell type specific chromatin regulation. And then there's the challenge of genome topology. And so even if we can identify what's called a cis regulatory element, so an element that's next to the gene, a DHS site that's like right next to the gene, that's not always actually how the genome is actually physically connected. And so it could turn out that a DHS site distally may be impacting a gene far away. And so I'll give you sort of a cartoon of kind of like how we're approaching this and how we're thinking about this. And so this is sort of the cartoon here where you have a GWAS SNP, so a single nucleotide change in a DHS, or in a site of open chromatin. And that single nucleotide change can affect the affinity for the transcription factor, or it can change the motif for the transcription factor, so the transcription factor no longer binds there, or maybe it induces a transcription factor binding site. And then that is what then, of course, drives active gene transcription. And so going back, man, maybe like 7 years ago, we developed a program where we started to think about a framework for how could we actually figure out these GWAS SNPs, like how can we link those to genes, right? Eventually to genes. And so we developed a program where we did what's called ATAC sequencing. This is the assay for transposase accessible chromatin regions. So this is looking at regions of open chromatin. Of course, you want to know what the readout is. The readout is, of course, gene transcription, and so you can assess that through RNA-seq. And then sometimes there can be, again, the genome folds on itself, and it doesn't do this randomly. It does this in a coordinated way. And so you'd like to know what enhancer elements or what regulatory elements are physically connected with what promoters. And so you can assay that with a technique called Hi-C. And so we developed a program led by Junaid and Paul, who are both MSTP students in the lab, in which we tried to do this to understand GWAS SNPs in a cell type specific manner. And so we took human coronary arteries. There were 44 that underwent a sequencing platform called the Multiome platform from 10x. And so these are single nuclei that are extracted from the tissue. And then there's a barcode that's assigned to each nucleus that's the same barcode. And there's a sequencing library that goes for ATAC, and there's a sequencing library that goes for RNA. And so then you can understand what areas of open chromatin are accessible in a specific nucleus and what is its impact on gene transcription. And then we took a subset of those, so 16 coronaries, and we performed what's called Hi-C to understand the physical linking between the two. As you can imagine, this is a large and expensive data set to generate. There's a funny and very informative story about how the Hi-C got paid for, but I'm not going to get into that here. And for the next hour, I'll tell you about the cell type specific map that we that Paul and Junaid had generated. But I'll leave a QR code if anybody's interested in the details. But this is the last data slide that I have, I think. But suffice it to say, we're excited about the fact that we've now been able to take, in a cell type specific manner, coronary disease GWAS variants, link those to enhancers, and then link those to either proximal or distal genes, again, in a cell type specific manner, giving an immediate hypothesis of how we should be targeting these genes in what cell types. So I'll wrap it up in time for questions. Hopefully I've convinced you that human genetic studies provide a unique opportunity to both identify and validate therapeutic targets in novel causal pathways. Hopefully I've convinced you that SVEP causally promotes vascular disease. We think that this is true using both human genetics and mouse models. We've shown that SVEP is the first known biological ligand of this receptor pair 1. We think that this is beneficial. If it will be beneficial is yet to be shown, but we think that targeting this is going to be beneficial. And we think that tissue multiomics is a really exciting way to link GWAS variants to genes in a cell type specific manner. And again, I showed this at the beginning. I'll emphasize the fact that these are the people that are incredibly passionate and creative and really a joy to work with every day, who actually do the work. And so I feel very fortunate to have such a great team. And then because I'm in my home court, I'll give a shout out to people. This is a woefully inaccurate and incomplete catalog of people that have been important to me as I first came here. And so, you know, WashU is really an incredible place, of course, with lots of great physician scientists and great mentors. And so these are some of the people that have been important. I'll highlight Doug Mann and Gene Narbonne. So Doug was the chief when I first showed up, took a flyer on an unproven guy from the East Coast who was a month into his K, I think. And then Gene was the head of the CCR when I first showed up, and those are, like, of course, really informative years about thinking about your lab and how you're going to set it up. And Doug and Gene were both really instrumental in sort of encouraging me to keep my head up and do the right science, and they've really encouraged me that no matter what people tell you, like, the truth really does matter, and so pursue the truth. And then, of course, there's a huge support staff, right? This is, again, woefully inadequate. These are some of the people that I worked with as a junior person who helped pay the bills and get the grants and manage the grants and wash the dishes and order stuff and, you know, keep the microscopes running. And so I will just say that this is inadequate, but WashU is a special place with lots of great people. I'll thank the collaborators. I think I've tried to show that this work that we do is super highly collaborative, both with people at WashU, people across the country that we've been working with for ANGPTL3 and this multiome project that I mentioned. And then, you know, human genetics is a team sport, and so there's lots of different cohort collaborations. And then, of course, I'll thank the funders, including the Longer Life Foundation, which has been really important. And I think that this is what you've all been waiting for.
Thanks ever so much, Nate. That was an absolutely fascinating talk. We are open for questions.
Okay, so that's a great question. So there's a question for those of you playing from home. There's a question, does SVEP relate to longevity? Mark, it's like you may have read this review that we wrote. So it turns out that it is. And the two pieces of data that I have, well, there's an indirect piece of data and then there's well, both are indirect. How about that? So there's a paper that measured 5,000 plasma proteins in a cohort, and they measured them at different age ranges. And they asked not only what proteins increase with age, but what proteins accelerate with age. It turns out that SVEP is the #1 protein that accelerates with age. So it not only increases, but the rate of increase increases with age. And so that's a function of age. And then the other one, it's of course hard to get to the real question of longevity. As you can imagine, they're long studies. But the UK Biobank looked at, sorry, it wasn't UK Biobank, it was a separate population cohort. They measured plasma levels, and they asked, what was the age of your parent when your parent died? And it turns out that higher levels of SVEP relate to younger parental death. And because it's heritable, or some degree it's heritable, it sort of gives an indirect indication that it is related to longevity. Yeah, it's an interesting question. We're trying to address this with the mice, but these studies are long. Yeah, so the question is the SVEP-pair interaction, and if SVEP is circulating, what regulates that levels, and is it pair? Is that a fair summary of? So we don't actually know how SVEP gets into the plasma. This is an extracellular matrix protein. It's made by stromal cells in tissues. Lots of ECM proteins get out into the plasma, like collagen, fibrin, all these sort of proteins. And so we think, we don't know, but we think generically that that plasma levels reflect tissue levels. And so higher tissue levels equal higher plasma levels. But we don't fully understand what regulates the levels in the plasma. We've looked, and actually pair levels don't really change that much. But what we think is happening is that, again, maybe with age, and certainly with insult, so with inflammatory stimuli, for example, SVEP expression goes up. And so we think that this is almost a feedback loop. I think, without a lot of data, that it may be an adaptive maladaptive response. And so in response to initial injury of the arterial wall, for example, this starts to get made, and then it starts to feedback on itself and increases leukocyte recruitment, which then increases inflammation and expression and sort of this kind of thing.
Terrific talk. I'm going to ask you a question that I think you probably would have asked one of our MSP applicants.
Uh-oh.
That is, what are your thoughts on actually the design of a clinical trial to test your inhibitor preventing coronary?
Right, so the question is, what are the thoughts about designing a clinical trial for the inhibitor? So we're trying to pursue this in animal studies, of course. I think the challenge that we have is we don't have this is the challenge, I think, with any therapeutic that's going to target the arterial wall. We don't have a great biomarker of target engagement. And that's, I think, what we're really lacking in terms of how do we know how to dose this? How do we know what frequency, et cetera? We can measure, there are ways, of course, to measure it in the plasma, but what we'd really like to do is know what it is in the tissue, and we don't have a great biomarker of target engagement. And so that's, I think, what we're lacking before we can actually move to those trials. We're going to try and pursue this in preclinical models and sort of see, but we've got some work to do, I think, before we can really think about a clinical trial.
And to follow up that question, are you thinking chronic use in people who are susceptible to disease, or maybe just acutely using it during a marked life?
Right, so the question is, would this be a chronic, or would this be sort of an acute onset issue? In unpublished data, we have some suggestion that acutely this might actually worsen things. So like, for example, treating an acute myocardial infarction with this actually may make things worse. Again, that may fit with what I mentioned before, where if it truly is like an adaptive maladaptive response, you would probably want to treat this after the initial insult, and then the duration and the chronicity is, of course, open for we don't know. Yeah. Oh, there's a question up here.
Sorry.
Yeah.
So in the preclinical models, do you need sorry, I didn't do my PhD in cardiovascular anything. Do you need
Most people haven't, it's okay.
do you need mice that are prone to like atherosclerotic disease?
Yeah.
Already?
That's a great question. So the question was, in preclinical models, do you need to have an animal that is predisposed to developing atherosclerosis? Yes, you do, for sure. So animals like mice, I actually don't know, other than primates, I'm not sure there are animals that develop atherosclerosis, to be totally honest. Is that true?
I think dogs get it.
Dogs, maybe? But certainly mice do not. You really have to try hard to get them to develop atherosclerosis. The typical way is by removing either the ApoE receptor or the LDL receptor and then giving them a high-fat diet to drive cholesterol very high. And that will induce this atherosclerotic plaque. Okay. Thanks, everybody.
Wonderful talk.
Oh, thank you. Thank you so much. Thank you. I appreciate that.
Quick question for you. So I may have missed this, but it looked like when you were looking at your SNPs and SVEP, you also had a signal for type 2 diabetes.